what happens. The cells grow so fast, so rapidly, it breaches the epithelial layer and becomes a big tumor mass. So this is a big malignant tumor that has arisen as a result of this particular mutation. What we need to do in order to be able to understand cancer is to be able to distinguish this red mutation from the blacks and the whites. If we understand that this is the red mutation that gives the huge growth advantage and it becomes a malignant tumor, then we have made an understanding, or we have an in-depth understanding of the tumor process. Again, even after this mutation has arisen, there are other mutations that keep arising. So as you can see, more mutations are happening, but again, these are innocuous mutations, they don't cause any harm. So two things. One is, in the background, there are mutations happening that are usually harmless, and some mutations happen that are hugely harmful. So we need to be able to identify those mutations that are harmful and weed out those mutations that are harmless. Okay? So this is what the research in cancer is all about. What are the genes and genomic alterations that drive cells to a growth advantage, eventually forming a malignant tumor? So it provides the cells with a growth advantage. So this is what we've been involved with. My own work in the last few years has been in the area of oral cancer, and oral cancer happens to be very important in India, especially among men, and so my last story was going to be saving lives of men. So that's called a driver mutation, and we want to identify these mutations that are called driver mutations because they drive the cell to becoming a cancer. So this is technically called squamous cell carcinoma of the oral cavity. It's oral cancer. 8th most common cancer. About 250,000 new cases arise annually throughout the world. 2/3 of them arise in developing countries. About 125,000 deaths occur annually. And it accounts for 1/3 of all tobacco-related cancers in India. And between 20 and 60% of cancer is associated with a virus that's called the human papilloma virus. The human papilloma virus is a very potent carcinogen, causes cancer. It causes 100% of cervical cancers. Cancer of the cervix is associated with human papilloma virus. The hazardous combination, we all know, khoini and gutka and all of this, tobacco, snake lime. The lime is chewing up the epithelial layer. The carcinogens in the tobacco are getting to the cells directly, and then that hazardous combination is what precipitates the mutations, and those mutations then cause cancer. So we're researching on this, and I'm going to share some information on patients. Most are male. This is predominantly a male cancer. Oral cancer is predominantly a male cancer. Most individuals are middle-aged. They present themselves at a very late stage. Usually what happens is that before you get cancer, you get white patches inside the mouth, but you think that these are just white patches, perhaps they will go away, so we don't care, we don't go to the doctor, but eventually when it becomes a big bruise, it becomes it's not looking good, it's becoming more and more, it's spreading more and more widely, then we go to the doctor, and then it's usually quite late. So most patients actually present themselves at quite a late stage. Most patients in our study were exposed to tobacco. About 80% of them have been using tobacco, mostly chewing tobacco, for over 10 years. About 25% of them have HPV infection. So what we did was, again, we did genome sequencing. So we did whole genome sequencing, meaning that we looked at DNA of these individuals, and we looked at the blood and the tumor DNA. Remember, in order to be able to find those changes, those DNA changes that happen during the life course of an individual, those will be present in the tumor DNA, in the cells of the tumor. And the ones that we are born with will be present in the blood. So imagine, if you will, that we get a list of changes that are present, list of cellular changes in the DNA that are present in cells of the tumor tissue, and we make another list of those changes that are present in the blood, and if you subtract one from the other, since the blood changes have been inherited from your parents, and the tumor changes are a mixture of those changes that you were born with, plus the additional changes that happen in your lifetime, if you subtract this list from this list, you get those changes that happen during the lifetime of that individual. So this is what we've been doing. We've been sequencing both blood and tumor DNA and trying to understand what are those DNA changes that drive. We've done, and once you get some discovery, you need to validate that discovery, and that's the process of science. So in science, you do some experimentation, you have a study design, you do an experimentation, you discover something, but then in biology we are forced, it's not physics, so in biology we are forced to again go and find out whether this happens in an independent set of individuals, and that's called validation. So validation of some of discoveries is a process of biological science. It's probably not a part of the process of physical science, but in biology we do this all the time. So the question that we asked is, what are the genes that drive, and what are the natures of these genes? I'm not going to tell you a long story, because obviously the story is long and technical, but please remember that there are two kinds, and I've told you this before, that there are these two different kinds of genes. One's called the tumor suppressor, the other is called the oncogenes. The tumor suppressors are usually awake, and a DNA change puts them to sleep. And once they go to sleep, they don't perform their normal activity, and that non-performance of that normal activity leads to cancer. The oncogenes are usually asleep, a DNA change wakes them up, and once they wake up, they produce terrible proteins, and those proteins lead to cancer. So we'll remember this. What we found in oral cancer is that it is predominantly the tumor suppressors that drive oral cancer. And the genes, again, for those of you who are from school probably wouldn't know the names of these genes, and those of you who are from a non-biological background wouldn't know the names of these genes, but the biologist in the audience would obviously readily recognize genes such as TP53. It's called the guardian of the genome. It's a very, very important gene. Caspase 8 and so on and so forth. The point that I'm trying to make is that even though there are a couple of oncogenes, most of them are tumor suppressors. And tumor suppressors is bad news. Because once a gene goes to sleep, it's very difficult to wake up that gene using drugs. When a gene activates itself, you can find an inhibitor to that particular protein that it is producing, to the rogue protein. But once it was producing a protein, it has gone to sleep, it's very difficult to wake up that gene to produce that protein. So when a cancer is due to tumor suppressors, that's bad news. We'll remember that, I'm going to skip a few. We'll remember that that's bad news, and I want to come back to this particular two individuals. One to ask the question, why is this individual smiling? This individual is smiling because in 15 days at a particular dose of a treatment, this individual completely got cured of cancer. There are no remnants of tumor tissue in the body, while this individual who received the same treatment for the same duration at the same dose, no change in 15 days. We will recall a gene, we will recall the name of a gene that's called BRAF. BRAF, mutations in BRAF, DNA changes in this particular gene are found in various cancers, predominantly in malignant melanoma, but also in cancer. 70% of malignant melanoma has changes in this particular gene called BRAF, because of which it drives the cell to becoming melanoma, and 70% of malignant melanoma is because of driver mutations in BRAF. Mutations in BRAF are also associated with 30% of thyroid cancer and 10% of colorectal cancer. So this is an important gene, and we need to understand, well, we've understood that there are mutations in these genes that drive to various forms of cancer. We need to understand what this gene does. So this is what the gene does. The gene produces a protein, the BRAF protein, and this is again what this protein does is to regulate signaling from outside of the cell to inside of the cell. So this is what BRAF does. So it's taking signals from outside of the cell and pushing it to inside of the cell. And one of the messages that it pushes to inside of the cell is either to ask it to grow, or at a certain stage ask it not to grow. And please remember that the hallmark of cancer is unregulated cell growth. So if something happens at the cell, the signal doesn't produce a signal to asking the cell to stop growing, it will just grow and grow and grow, and then it will form a malignant tumor. So this is, like I said, this protein is involved in sending signals inside cells which are involved in directing cell growth. So if there is a mutation in this particular in this particular gene, so this is a BRAF mutated, it is not actually producing its proper function of taking signals from outside of the cell to inside of the cell and asking the cell to stop growing when it doesn't have to grow. So the cell just keeps getting the signal grow and grow because this is a mutated protein and it's not performing its proper function, and the cell just keeps growing. And as a result of that, the cell gets a huge growth advantage. It becomes a malignant tumor. One way to stop this is to be able to find a blocker of this mutated protein. So when it's mutated, you want to block it. But when it is not mutated, you don't want to block it. So you want to find a molecule that will block this particular protein only when the BRAF is mutated. Well, BRAF can be mutated in multiple places, and it's not easy to find molecules that will act against a variety of mutations. Fortunately, BRAF has one common mutation, and the medicine that has been found, the molecule, the drug that has been discovered, it acts against that common mutation and therefore is very, very helpful. So that's a drug that's been found, and essentially what that drug does is to block that protein. And this BRAF protein that is the common mutation, like I said, is at a particular position of the gene, and there's a drug that has been found which is an inhibitor, and this acts as an inhibitor only if this common mutation is present. And because this mutation is common, it is helpful in the vast majority of patients. It's called Vemurafenib, and Vemurafenib has changed lives of people who actually have a BRAF mutation and have different kinds of cancer. So as you can see that the same gene is involved in multiple cancer types, so there may be a day when you really don't need to know what kind of cancer you have. All that you need to know is what kind of gene is mutated that has caused cancer in you. And once you know what kind of a gene it is, if you can find an inhibitor to the gene or some other ways of ameliorating the ill effects of that gene, then you are done. So this is what has happened. This individual, it's personalized treatment depending on whether the individual has a mutated BRAF or not, the V600E mutation or not. This individual did have that mutation, as you can see. So this individual had a BRAF mutation. Vemurafenib was used, and this individual responded very, very well and almost became free of cancer. While this individual did not have the BRAF mutation, Vemurafenib acts only, that drug acts only when there is a only against a mutated protein. So if there is no mutation, it does not work, and therefore this individual did not even respond. So it's important for us to know what kind of mutations are there in that particular individual to be able to use this particular drug and hope that this individual will respond. So this is the knowledge of genetics genetics and genomics is actually helping find proper treatment and also discover proper drugs. I'm going to skip this. The last point that I want to make is with respect to movement of or having multiple tumor types within the body. It's called a process of metastasis. This process of metastasis is like if you have lung cancer, you also have after some time brain cancer. So you treat for lung cancer, and then after a certain time you have brain cancer. So these cells from the tumor tissue in the lung have migrated through the blood and have breached the blood-brain barrier and has reached the brain, and you have brain cancer. So similarly, breast cancer can lead to other kinds of liver cancer and so on and so forth. So one organ is affected, that particular organ sheds these cells, and the cells migrate through the bloodstream and goes and homes into another organ, and then that organ gets becomes malignant, and this whole process is called metastasis. What we try to understand is that in oral cancer, the kind of cancer that we do research on, the metastasis happens in the lymph node. So even though oral cavity is affected, the tumor cells from the oral cavity will go and lodge itself in the lymph node, in the near lymph node, and the lymph node there will be metastasis in the lymph node. There will be cancer in the lymph node, and that's a metastatic cancer. So in our data, we found 21% of the patients who had lymph node positive, meaning that they had cancer in the lymph node, and 2% of our oral cancer patients did not have no, I'm sorry, I'm sorry, I'm completely withdraw what I said. These percentages are something different. So we had a fraction of patients who had lymph node metastasis, who had cancer in the lymph node. Their primary site was oral, their secondary site was lymph node, and we had a bunch of patients who had oral cancer, but even after a number of years did not have lymph node metastasis. So we had a bunch of patients in whom there was metastasis and a bunch of patients in whom there was no metastasis. And what we asked is, can we find genes that drive this process of metastasis? Again, to cut a long story short, what we found was that there are mutations in two genes. One's called BRCA2. Remember BRCA2? Produces ovarian cancer and breast cancer. And remember Angelina Jolie? So that's a connection, but it also has a connection with metastasis. So there are mutations in BRCA2 that lead to metastasis in oral cancer, and the other gene is FAT1. And what we find is that in the lymph node positive, in the metastatic lymph node cases, 21% of these genes are mutated, and in the lymph node negative cases where there is no metastasis, only 2%. So 2% is like the baseline, and over the baseline, about 10-fold greater chance of metastasis if you had mutations in BRCA2 or FAT1. Come to the last part of my talk, which is to wrap up this conversation. Remember the conversation, the doctor tells the wife that I need to put him on chemotherapy. The wife panics, says, can I take him home and administer chemotherapy at home? The doctor says, no, let me check out his EGFR mutations. If he has mutations, you can take him home. This is wonderful because now the treatment is tailor-made towards having or not having mutations in this particular gene called epidermal growth factor receptor. So epidermal growth factor receptor has, I'm not going to get into the details, but what happens is that if there are changes in this gene, it eventually leads to uncontrolled cell proliferation. And uncontrolled cell proliferation is a hallmark of cancer that I've been repeating many times. Again, mutations here lead to this. One drug that was available or that came into the market is called afatinib. This drug is a selective, orally bioavailable blocker of EGFR, mutant EGFR. So that's the reason why the doctor said that if you have mutations in EGFR, you can take him home because this is orally bioavailable. Because then you can use oral chemotherapy and not injectable chemotherapy. So there are drugs that have been discovered that will work against a mutated EGFR, and the response rate, so this is an oral which is called afatinib, and injectable one is called cisplatin. This is a platinum-derived drug molecule. The oral, not only if you have EGFR mutations, not only can you use oral chemotherapy and dispense with injectable chemotherapy, but also the oral chemo to such depths that we will be able to precisely get medication for different individuals. So this is what our dream is: unleash the power of genomics to effect precision medicine and turn cancer into a chronic disease. 30 seconds, let me tell you what this means. Today I'm hypertensive, and what I do in the morning is after breakfast I take a pill which keeps my blood pressure under control for 24 hours. So I take my antihypertensive pill again tomorrow at immediately after breakfast. So I'm not cured of hypertension. Hypertension remains, high blood pressure remains. But what I can do is to take a drug and keep it under check such that I'm chronically hypertensive, but my blood pressure is under check. So today, most cancer patients die. And if you can find, through this genomics approaches, if you can find a pill, remember the oral chemotherapy, that's really a pill. If you can find pills where, if you have cancer, you just take a pill in the morning and go about doing your daily activities, and you take a pill the next morning and go about doing your daily activities, then you have turned cancer into a chronic disease, just as nobody now dies of hypertension. They just take a pill in the morning and are able to go through their daily lives in a normal way. So what our dream is to be able to use all of this genomic information in order to make medicine, practice of medicine more precise, such that even a disease, a dreadful disease such as cancer, will become a chronic disease. Thank you very much for your attention.
cancer, and the cell migrates to the brain. So brain has a completely different environment. So is it the same set of genes that are expressed or sort of mutated in lung? Are the same set of genes are also mutated or in the brain's tissues?
So when the cell is migrating and lodging itself in another tissue, whether it's brain or bone or wherever, it's carrying that set of mutations. Then probably another set of mutations through the life course is happening. That's again precipitating a new set of mutations can also happen. But usually it's that set of mutations, because it has already changed certain proteins and made certain proteins faulty, and that cell is going there. So it's likely that it is the same set of mutations that is also causing cancer in the other tissue. The question that we are asking is, why is it that not everybody produces metastatic disease? If you have same cancer types, if you take 10 people, maybe 2 of them produce metastasis, the remaining 8 do not produce metastatic disease. So the question is, why is it that these 2 people produce, is it just a random chance? Or are there genes that drive the dislodging of cells from the primary tissue for it to migrate through the sail through the bloodstream and go and lodge itself in another organ? So there is, again, I don't have the time to get into the technical details, but there is room to believe that there are genes that are involved in this process of dislodging, migrating and homing into another organ. And we found 2 in oral cancer that's involved in the process of lymph node metastasis, BRCA2 and FAT1.
Okay. And quick question is, essentially, is there any role of mechanical stress inducing the mutations, for example, in the primary mutations?
Mechanical stress inducing mutation.
For example, what we know is that the genes expressions are a function of the mechanical stress. For example, if a mechanical stress is given to the certain in the nucleus, for example, then certain genes are expressed, or so is there.
So by mechanical stress, what would you mean? Physical stress?
Yes.
How do you induce physical stress to the nucleus?
So in some in vitro experiments are done, sort of, for example, pulling the cells, so which essentially triggers certain genes in expression.
So this is possible, but I would imagine now I don't know much about it, but I'm imagining that cannot be a common way of producing mutations. It may happen, but I don't think that's a common way. But again, I may be completely wrong. So I don't know. What I do